Hello everyone, welcome back to another video. In the previous video you learned a bit about synthetic biology and its purpose. We will now move on to methods of synthetic biology. The first method we will dive into is antibiotic resistance. Antibiotic resistance is the ability of bacteria to safeguard themselves from the effects of an antibiotic. When in the presence of an antibiotic, the bacteria will survive or multiply faster than bacteria that is susceptible to the antibiotic. The two pathways that bacteria have in order to obtain resistance are mutations and the receival of resistance genes. Mutations are random changes in the DNA of the bacteria that may possibly provide resistance. Receiving resistance genes from nearby bacteria is a process known as horizontal gene transfer. Vertical gene transfer is the inheritance of these genes in new generations. There are numerous routes that this bacteria can spread through, including humans, animals, and the environment. Here is a helpful chart. Horizontal gene transfer can occur in both bacteria of the same species and bacteria of different species. There are three significant ways for bacteria to share their genes. Conjugation is when two bacteria connect through structures in the cell membrane and transfer DNA from one cell to the other. Transduction is a process involving viruses called bacteriophages. These viruses can infect bacteria. They pick up genes from other bacteria they infected and incorporate them into their new host. Transformation is when the bacteria takes pieces of DNA from directly around the cell. All genes, including antibiotic resistance genes, can be transferred between DNA. The question is whether or not the gene will be consolidated into the DNA. There are machineries in place that will work against this incoming DNA, however, they are not completely efficient. How do scientists track the spread of these genes? This brings us to GFP, an extremely important protein in scientific research. GFP is a green fluorescent protein. It is found in jellyfish from the North Pacific. It absorbs UV rays from the sun and releases it as green light. This protein can be attached to anything that needs to be watched. For example, a scientist can attach it to a virus. To track the spread and growth of the virus, all the scientist would need to do is shine an ultraviolet light on the sample. GFP will shine green and allow for the scientist to record the data. Similarly, GFP can be used to track the spread of genes that can make bacteria antibiotic resistant. Let's recap. Some bacteria can become antibiotic resistant and spread quickly as a result. This can be a result of either mutations or the receival of resistance genes. Resistance can be spread many ways, including conjugation, transduction, and transformation, which are the three main ways that bacteria can share their genes. All genes can be transferred, but there is no guarantee that they will be included into the DNA. GFP helps scientists track the transformation of resistant genes in bacteria. That's it. Thanks for watching. In the description there are the sources that we used in this video. Thanks again.